Whether you are examining the intestines or the bladder, the failure of visceral organs follows a universal, highly predictable 10-step sequence of degradation. The foundation of cellular biology relies on a strict thermodynamic axiom. Structural order requires a continuous supply of adenosine triphosphate, or ATP. ATP is the primary energy carrier in all living organisms. Pathology begins the exact millisecond that cellular energy production drops. This diagram illustrates what is known as the universal cell death memory rule. It states a strict thermodynamic difference. Orderly, programmed cell death requires energy to execute, whereas chaotic, destructive cell death happens when that energy is entirely gone. This raises a clear clinical question. How do completely divergent medical emergencies, like an ischemic bowel on one hand and acute urinary retention on the other, stem from the exact same molecular failure? The intracellular mechanism of failure is identical across these tissues. The drastically different clinical consequences we see in the emergency room are dictated entirely by the unique native functions of the specific organ. Consider the gastrointestinal tract. Its baseline energy requirements are massive, constantly fueling nutrient absorption, fluid secretion, and maintaining a strict immune interface with billions of gut microbes. The genitourinary system has completely distinct energy demands. It expends ATP to store and void urine, maintain smooth muscle tone, and uphold a highly impermeable barrier against toxic waste. This graphic shows step 1, the initial insult. For the GI tract, this is triggered by ischemia, shock, or sepsis, a systemic inflammatory response. Meanwhile, the GU system faces distinct initial insults, including an obstructed ureter or severe urothelial infection. Despite these radically different macroscopic triggers, the biological destination for both tissues is exactly the same: an inescapable cellular energy crisis. That crisis is step 2. As blood flow, and therefore oxygen, decreases, a state called hypoperfusion, there is a rapid and critical fall in intracellular ATP production. The GI mucosa is exquisitely sensitive to this sudden energy drop. Its cellular turnover is fast, and its metabolic demand is constant. A parallel effect occurs in the GU system. As smooth muscle energy depletes, the active transport mechanisms of the urothelium immediately begin to degrade. This brings us to step 3, pump failure. Loss of ATP removes fuel for active transport, completely halting sodium-potassium pumps and causing a rapid buildup of intracellular sodium ions. This sudden accumulation creates a powerful osmotic gradient, aggressively pulling surrounding water into the cell. The physical consequence of this water influx is rapid and severe cellular swelling. This swelling forces distinct structural failures. In the gut, the physical expansion causes tight junctions between cells to pull apart and weaken. In the bladder, the exact same swelling critically destabilizes the internal ion gradients required for muscle control. The initial microscopic loss of energy has translated directly into a mechanical failure. The cell has lost the ability to regulate its own volume and maintain its structural boundaries. The danger escalates in step 4, calcium dysregulation. This marks the transition from mere volume failure to active cytoskeletal damage. Without energy to pump it out, intracellular calcium levels spike. In the GI enterocyte, this calcium actively damages the junctional proteins holding the mucosal lining together. In the GU tract, this same calcium spike dysregulates smooth muscle handling, causing impaired, erratic contraction of the bladder wall. The cascade continues into step 5, reactive oxygen species, or ROS. Ischemia reperfusion and recruited neutrophils flood the compromised system with these destructive molecules. The specific effect of this ROS flood is severe lipid membrane damage. This systematically shreds the outer boundaries of the cells, critically worsening permeability across both tissue types. The cellular environment becomes increasingly toxic in step 6, acidosis and barrier failure. Localized metabolic byproducts now begin to compound the existing structural damage. Ongoing blood flow failure forces cells to generate energy anaerobically, producing lactic acid. This local lactic acidosis leads directly to cellular ulceration, rendering the mucosa massively leaky. The intracellular chaos has now reached the organ level. The foundational cell membrane is broken, setting the stage for devastating systemic exposure. This exposure triggers step 7, Inflammation. The body detects the compromised cellular barriers and launches a systemic immune response. Tissues release cytokines, specifically IL-1, IL-6, and TNF-alpha. These signaling proteins trigger massive neutrophil recruitment and activate the endothelial lining of blood vessels to mobilize immune cells. The localized consequences of this cytokine storm are severe. Tissues swell with profound edema, which in the bladder manifests as intense pain signaling. In the GI tract, the resulting inflammation creates a pathway for endotoxins, toxins present inside bacterial cells, to catastrophically enter the bloodstream. The body tries to save itself in step 8, compensation. During shock, the autonomic nervous system plays a destructive role. It forces splanchnic blood flow, feeding the abdominal organs, to aggressively constrict, redirecting blood to the heart and brain. While preserving the brain short-term, this actively starves the GI tract, worsening gut ischemia. A similar paradox occurs in the GU system. Autonomic nervous system shifts alter bladder outlet resistance and internal pressure, which only compounds the local muscular dysfunction. The body's own desperate systemic survival mechanisms inadvertently accelerate and guarantee the localized tissue's destruction. The microscopic 8-step cascade finally manifests as a macroscopic clinical emergency in step 9, organ failure pattern. As illustrated here for the GU system, these clinical endpoints include acute urinary retention, poor emptying, and severe ischemic mucosal injury. Contrast this with the GI tract systemic endpoints. Patients present with profound ileus, an inability of the intestine to contract normally, along with severe malabsorption and massive loss of the intestinal barrier. The diagram notes a special pearl for this exact moment. This barrier loss yields a unique and deadly consequence: massive bacterial translocation, where gut microbes directly invade the bloodstream. Identical cellular energy failures have now produced radically different clinical threats. We see a mechanical blockage in the bladder versus a systemic infection originating from the gut, based entirely on the organ's native anatomy. Finally, we reach step 10, cell fate. The ultimate outcome for the tissue depends strictly on the severity and duration of the initial ischemic insult. Recall the universal cell death memory rule from earlier. It explains the first possible cellular fate, apoptosis. Apoptosis is a highly regulated and programmed process of cell death. If the cell retains a baseline level of residual ATP, it executes this orderly death. It cleanly packs itself away into fragments without damaging surrounding tissue. Contrast this with necrosis. If severe ischemia totally depletes ATP, the cell chaotically ruptures, spilling its contents into the local environment. This chaotic, uncontained rupturing is the direct microscopic cause of macroscopic catastrophes, leading directly to events like an acute bowel perforation or severe urothelial shedding. From the initial insult to total cell death, the 10-step ischemic cascade is an inescapable, continuous biological process. The clinical divergence is clear. GI failure primarily presents as a barrier breach that quickly leads to systemic sepsis amplification. GU failure, on the other hand, presents as mechanical dysfunction, marked by altered pressure and hazardous fluid retention. Yet, despite these vast macroscopic clinical differences, the stepwise pathophysiology of all visceral organ failure is completely identical. Mastering this homologous pathway empowers clinicians to look past divergent clinical symptoms. It allows them to trace complex systemic failures back to a singular, treatable root cause: cellular energy depletion.